Okay, so hello everyone. This is the biology periodic in week 10. Okay, I will start with the MCQs first, okay, since it's going to be easier for me. So I'll just go through all the MCQs first and then I'll begin with the FRQs. Now I will be posting one of the FRQs later, which is number 14. Okay, this question I'm not going to post it later because it's not gonna come. So, like, to be very honest with you, like, I don't understand the question yet, but I am going to try and make sense of the question. If anyone actually knows the question, please let me know and like explain it to me so I can actually explain it to everyone else, because I genuinely don't understand the question yet, but I will try and make sense of it, and when I do I will help you guys with it. Okay, so let's start with the MCQs. So this was an AMS question, it came in the last AMS. So the illustration shows the active transport of hydrogen ions. Which of the following best predicts the effect of not having ATP available to supply the energy to this process? Okay, so as you can see here, we have a low concentration of H plus ions and then a high concentration of H plus ions. So we're going to need ATP to actively transport it from an area of low concentration to high concentration. Okay, so we're going to take the H plus and put it here, which that isn't supposed to happen, but since we have ATP, we're going to have active transport, so that happens. Now, when we don't have ATP anymore, when this is gone, it doesn't necessarily mean that this is going to go here. That doesn't mean that doesn't necessarily happen. It might, but it doesn't necessarily happen. We don't know that it will happen. So we're going to pick the option that says the H plus ions will stop moving through the protein. Everything will just stop. Okay? Now, number 5. Which of the following transport mechanisms will be affected most directly by a temporary shortage of ATP? So remember, ATP is the thing that helps active transport actually happen. And active transport is the transport of something from a low concentration to a high concentration. So, which means that it's going against the concentration gradient. So if we don't have ATP, it can't do that. So that means it will greatly affect the transport of glucose molecules against the concentration gradient, because without ATP, that won't be able to happen. Okay, so this one, like, just focus with me here. Okay, so based on the model represented, which of the following changes will most likely represent a depletion of available ATP? Okay, so basically this is the plasma membrane here. You have your bilayer, you have these three transport proteins, okay? You have here a high concentration of Na and a low concentration of K+, and here a low concentration of Na and a high concentration of Na of K+. So, as you can see, we have K+ leaking out. If here, so we'll look at it one by one. So K+ leaks out, okay? K+ has a high concentration here and a low concentration here. High to low is passive. That means no ATP required. So this will go on normally without ATP, because they're asking us what will happen without ATP. So this will go on normally without ATP. Then we're gonna go look at the Na. Here we have a high concentration of Na, and here we have a low concentration of Na. Here Na leaks in, so here to here it will happen. Okay? Normally, no need. But here, look. K+ is high here. Sorry, what's it called? Na+ is low here, but high here. See? High here, low here. So Na+ is going against its concentration gradient, meaning it needs ATP. So if we don't have ATP, the concentration of Na inside the cell, because this is the inside of the cell, will increase. Okay? So concentration of Na+ inside the cell will increase. Figure 1 shows the model of how a channeled protein influences the movement of a particle across a cell's membrane. Okay. An investigator wants to understand whether a newly found membrane protein is involved in membrane transport of a certain particle. Which investigation will determine whether the new membrane protein is a channel protein involved in the membrane transport? So, what we could do here is we'd add more of the proteins, okay, to the plasma membrane, and then we'd measure the rate of the movement of the particle. If we add more proteins and more particles are moving, that means that yes, this protein is involved in the transport of particles, because when we added more of the protein, more particles were transported. That means yes, it's involved in it. If not, then no. That's how you test if it is involved or not involved. Okay, so number 8. This is the general structure of an amino acid, the amine group, carboxyl group, R group, okay? Which of the following amino acid components shown in figure 1 best determines the interaction between an embedded protein and the hydrophobic regions of the cell membrane? The thing that gives it away here, hydrophobic regions, okay? When they say the hydrophobic regions or the hydrophilic regions, the thing that determines if it's hydrophilic or hydrophobic is the R group attached to it. If the R group is hydrophilic, then it's all hydrophilic. If it's hydrophobic, then it's all hydrophobic, okay? So this is what determines it, the R group. Okay, so the R group bonded to the central carbon. This one was in the other quiz, the BT101, the one for the last periodic. This one's BT102, so they just copy-pasted it in here. Which of the following provides evidence to best support the researcher's claim? The researcher's claim is the particular organelle originated from a free-living prokaryote, okay? And then it was later engulfed. So what shows us that something was engulfed if it has a double membrane? Now, which statement best describes the effect on water transport across the cell membrane if the aquaporin in the figure ceases to function? So first, let's see. So they're asking us, what happens if the aquaporin stops working? What does the aquaporin do aslan? The aquaporin takes a lot of water molecules, okay? And it transports it into the cell in bulk, okay? Without this aquaporin, water still transports by osmosis. It still transports by osmosis, but it transfers at a slower rate, okay? Not as fast as with the aquaporin, because the aquaporin will take a lot of it and put it in very quickly. But without the aquaporin, water can still move by osmosis, regular, just normal osmosis, but it will do it at a slower rate. So water molecules will be stable to move across the cell membrane, but at a slower rate. Number 11. Okay, so the ATPase requires the use of ATP to move ions from one side of a cell membrane to another. This is like the K plus, Na plus movements, you know. The amino acid cysteine is important to the function of ATPase, okay? So ATPase is the amino acid cysteine is important to the function of ATPase. Now, a change in the cysteine prevents the ATPase from functioning. Okay, so now the ATPase, which is the thing that is controlling the movement of ions from one side of the cell membrane to the other, okay, stops working. It stops functioning. Okay, why? Because the cysteine amino acid changed. The cysteine amino acid changed, the ATPase stopped functioning. What do you think will happen? The electrochemical gradient will change. Now, I know you guys, maybe some of you do know what an electrochemical gradient is. Some of you don't. You guys don't need to know it. You guys don't have to know it. All you have to know is when they say electrochemical gradient, they mean like ions. They're talking about like the ion gradient, like Na+, you know, like the normal gradient would be from high to low. That's all they're saying. Instead of just saying the regular gradient, they added a fancy word here, the electrochemical gradient. Obviously, if you can't transport ions anymore, the gradient will change, because then the areas of high concentration might not have high concentration anymore. Maybe it will become low concentration. You never know what can happen when you stop transporting ions. So when you stop transporting ions, the gradient gets messed up. Okay, here we have an experiment. So beetroot cells contain a family of dark red pigments called betalains. Wow. The selectively permeable nature of the beetroot cells keeps the internal environment of the cell separate from the external environment of the cell. Now, researchers are interested in determining whether the selective permeability of the beetroot cells is due to the cell membrane or the cell wall. So they want to see what's responsible for the selective permeability of my cell. Is it the cell wall of the cell or is it the cell membrane of the cell? Now, exposure to cellulase, here's the key thing. The exposure to cellulase is known to damage the structure of the cell wall. So when you put the beetroot cells inside cellulase, their cell wall starts to break. Okay? Now, an experiment is set up in which beetroot cells are placed in an aqueous solution with cellulase. So they put the cells in cellulase, okay? And in one without cellulase. So they put one with cellulase and one without cellulase. So they took two cells, one of the cells they put it in cellulase, one of the cells they put it no cellulase, okay? Now, which of the following results best refutes the alternative hypothesis that selective permeability is a consequence of the cell wall? So they want us to prove that the selective permeability is not a consequence of the cell wall. Okay, now what did we say that the beetroot cell's color was? It was red. Red pigments called betalains. See here, red pigments called betalains. So if the selective permeability of the cell is not due to the cell wall, that means it's due to the cell membrane. Meaning, if it's due to the cell membrane, the red stuff won't leak out even if the cell wall isn't there, because the cell wall ما دخل. The cell membrane is the thing that we're looking at. So if the cell wall isn't there, red pigments won't leak out. If the cell wall is there, كمان red pigments won't leak out. We don't care. We want the cell membrane to be responsible. That means even if we put it in cellulase, the solution that we put the cell in should remain clear. No red should be shown. So when beetroot cells are placed in a solution with cellulase, that means that their cell wall is destroyed. The solution remains clear, meaning that no red is spilled. Okay? Even without the cell wall present. Okay, the antibiotic Sidin S is a large molecule that can inhibit protein synthesis in mammalian cells. A researcher is investigating how S enters mammalian cells. The researcher identified 2 proteins that might be associated with S transport into the cell. So when we see something like this, when we see an antibiotic, okay, and they're telling us like it's a large molecule and I don't know what, and then they saw 2 proteins. They want to see what the function of these 2 proteins are. What's their role in all of this? What's their role in the transport of the cell, of the antibiotic? So obviously if we have proteins and then we have transport happening as well, and we have a large molecule, that means that those proteins are actually transport proteins most of the time. So that's why we say they are transport proteins that facilitate the movement of S across the cell membrane. Okay? I'm not gonna read the big fancy words. I'm sorry, but yeah. Which of the following best describes the relationship between the size and shape of a cell and the ability of the cell to move substances across the membrane? So, here what they went with is smaller irregularly shaped cells are more efficient at dissipating heat than large irregularly shaped cells. Why? Look, smaller cells have a higher surface area to volume ratio. And what did we say about surface area to volume ratio? Surface area to volume ratio, the higher the surface area to volume ratio, the more like Ya Allah, I lost the word. What? Smaller cells have a higher surface area to volume ratio, making them more efficient at heat dissipation and other membrane-dependent processes than larger cells. Shaped. Oh, صح. Okay, I just remembered it. The higher surface area to volume ratio allows the cell to exchange materials better. So the higher surface area to volume ratio, the better you are at exchanging materials. So the smaller the cell is, the higher the surface area to volume ratio. That means the better it is at exchanging materials and temperature, heat. The better it is at exchange, basically. Just not only the exchange of materials, just exchange in general. So the smaller it is, the higher surface area to volume ratio it is. That means it is more efficient at dissipating heat than the large irregularly shaped cells, because the large cell has a small surface area to volume ratio, so it's not as good as exchange. Number 16. So which of the following observations would best support the alternative hypothesis described above? So let's read. We want to support the alternate hypothesis. Plant cell walls are composed of cellulose, while fungal cell walls are composed of chitin. A group of scientists hypothesize that this difference means the cell wall has largely different functions in plant cells and fungal cells. Alternatively, another group of scientists hypothesize that despite their biochemical differences, plant and fungal cell walls serve similar functions. So what is the alternate hypothesis? Another group of scientists hypothesize that despite their biochemical differences, plant and fungal cell walls have similar functions. So we're trying to find the similarity here. And what's the only similarity they gave us? In both plant cells and fungal cells, the cell wall surrounds the outside of the cell membrane. That is the only similarity we have. That is why we choose this option. Which of the following observations best supports the model? So researchers have proposed a model of chloroplast evolution. According to the model, chloroplasts evolved from a small prokaryotic organism that was engulfed by an ancestral eukaryote. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host. So, what proves this? It's that both the chloroplast and the prokaryote actually share similar photosynthetic reactions, meaning they actually did originate from a common ancestor. And number 18. Okay, so here we have another experiment. Certain bacteria can use both ethyl alcohol and acetate as sources of nutrients. Okay, so bacteria can use ethyl alcohol and acetate as nutrients. In an experiment where both nutrients are available to a bacterial population, the following results were obtained and graphed. So we have the concentration outside the cells, so 0.1, 1, 10, 100, whatever. And then we have the rate of substance entry, 10, 100, 1000, okay? And what additional procedure would help determine whether these movements are due to active transport or passive transport? Okay, so basically what they did, they have a cell. Around it in its environment, you have acetate and ethyl alcohol, okay? Acetate, ethyl alcohol, okay? Around this cell, they put the concentration of it. So they put, let's say, مثلا, I don't know, 10 concentration of this and 10 concentration of that, okay? And then here they put the rate of entry. So the rate of entry, the rate of entry, the rate of entry. Let's say here the rate of entry, or whatever, 100 for the acetate, and then like, I don't know, 30 for the ethyl alcohol, whatever, it doesn't matter. They want to see whether these movements are due to active transport or passive transport. Now, what is the difference between active transport and passive transport? It's energy, ATP. That is the difference. Active transport needs ATP, passive transport doesn't need ATP. So what we can do is, we use two additional treatments, one containing only ethyl alcohol and one containing only acetate, and include a substance known to block ATP used by plasma membranes. Compare the graph of these two treatments to the original graph. Now, why did they do this? They used two additional treatments, one containing only ethyl alcohol and one containing only acetate. Now you're going to ask me, why don't they just mix it like in the original experiment? Because in the original experiment, they mixed it. They had acetate and ethyl alcohol in the same environment. Now, the reason they did this is because, what if acetate was due to passive transport, but ethyl alcohol was active transport? You don't know. That's why you separate them. That's number one. Now, number two, why did they add a substance to block ATP use? They block the ATP use to see, okay, if we block the ATP use and there is still transfer at relatively similar rates, that means it was always passive transport. But if we lower the ATP use and we stop getting the relatively similar transfer, that means it was active transport. That's why we do that experiment, we compare the graphs, meaning we compare the rates, and see, was it active transport or passive transport? And that is how we determine whether the movements are active or passive. And that's how you do it. So part D is the correct answer. Excuse me. So, a student is using dialysis bags to model the effects. Sorry. So a student is using dialysis bags to model the effects of changing solute concentrations on cells. The student places one dialysis bag that contains 25 milliliters of distilled water into each of two beakers that are filled with 200 milliliters of distilled water. Figure 1. The membrane of each dialysis bag membrane contains pores that allow small solutes, each as a monoatomic ions, to pass through, but are too small for anything larger to pass. Now, after 30 minutes, 5 ml of a concentrated solution of albumin, a medium-sized water-soluble protein, is added to one of the two beakers. Nothing is added to the other beaker. After 2 more hours at room temperature, the mass of each bag is determined. There is no change in mass of the dialysis bag in the beaker to which no albumin was added. Okay, so here they have a bag. Here they have a bag with pores, little tiny holes, okay, that things can transfer between. So like you can have osmosis, you can have like high water potential to low water potential, whatever. Okay, and then you have, what's it called? You just have distilled water, which is just, okay, water. Actually, you have like a beaker with water, and then this bag, okay, with the little pores. you have, and then now you're conducting an experiment and you're seeing and you're observing the mass. So without anything, the inside of the bag should have the same concentration as the outside of the bag. So we're fine, okay? So nothing is happening, nothing is like changing. So the mass is constant. So nothing will pour out of the bag or nothing will come inside of the bag, since the two concentrations are constant, since the two water potentials are the same. So we keep that constant, okay? So the mass should be a straight line. But then we added something, the protein. We started adding this protein. Now the water potential of the outside environment of the bag, which is just the water, the distilled water, now the water potential has decreased because we added the solute. That means to make everything balanced, we need to add water from the inside of the bag. So the inside of the bag starts to release water. But that means that the mass of the bag will decrease. So that's what our graph should look like. That is the graph. Okay? So which of the following best describes a cell that is likely to have an extensive Golgi complex? The extensive Golgi complex, what is aslan the function of Golgi complex? It is, what's it called? It's like packaging and modifying proteins, enzymes, whatever, okay? So a stomach cell that produces and releases digestive enzymes, that will definitely need an extensive Golgi complex. Which of the following statements best supports the claim that certain organelles within eukaryotic cells evolved from free-living prokaryotic cells? Okay, so supports the claim that certain organelles within us, the eukaryotic cells, evolved from free-living prokaryotic cells. Some organelles contain their own DNA that is more similar to prokaryotic DNA in structure and function than to eukaryotic DNA found in the cell's nucleus. So if I have something within me that is more similar to something else, that means that something within me was taken from something else. It originated from something else. That is why we say that we evolved from a common ancestor, which was a prokaryote. Moving on to question number 22. Oh my god. Okay. Are single cell eukaryotes that live in aquatic environments. The chloroplasts found inside are enveloped by three membranes, not two membranes, three membranes, as represented in figure 1. The inner membrane of chloroplasts resembled the thylakoid membrane. Okay, so this is the diagram, this is them showing us. Which of the following claims about the origin of the chloroplast is best supported by the three membrane structure of the protein? Okay, so we know that we know that a endosymbiotic event, meaning what do they mean by an endosymbiotic event? Means that one thing ate another thing and then it ended up looking like this because this ate that, so this surrounded this and it became that. So this surrounded this and it became, well, and then it started to look like this. That's one endosymbiotic event. But then an even bigger this came and ate that. So then it surrounded itself to that. So it lost that and it became something like that. So it had this. That means two endosymbiotic events, which means it originated from the incorporation of a photosynthetic prokaryote into a eukaryotic cell by two endosymbiotic events. Now, why a photosynthetic prokaryote? What are they telling you? Thylakoid membranes. What did they say here? أصلا they said chloroplast. Chloroplast على طول. Chloroplast, photosynthetic. No need to think about it. Chloroplast, photosynthetic. So two competing hypotheses exist regarding the cell membrane structure. One hypothesis states that membrane structure is static and membrane components throughout the bilayer are rigidly bound. Alternatively, the other hypothesis states that cell membranes are a fluid mosaic in which membrane components may drift within the bilayer around the surface of the cell. An experiment is set up in which the membrane proteins in two different cells are fluorescently labeled with two different colors and then fused as shown in figure 1. So basically they got these two proteins here together, they fused them together, and now they want to see what will happen. They are telling us which of the following results after 1 hour of the membrane fusion will best support the alternative hypothesis. What is the alternative hypothesis? That it's fluid mosaic. What does fluid mosaic mean? It means that they mix and match together. So this is cell 1 and this is cell 2, and that's protein 1 and this is protein 2. When they fuse together, if it's fluid mosaic, it should look something like this. Everything is mixed here, something dark here, something light here, something dark here, light here, dark, light, whatever, random هيك. Something like this. Everything is like smushed together. And as you can see, smushed together is the correct answer. See how they're smushed? One is dark, one is light, one is dark, one is light. That means it's fluid mosaic. They move, switch fluid inside. Okay. 24. Okay, so the fluid mosaic model of cell membranes describes proteins and steroids associated with the phospholipid bilayer. Excuse me. Which of the following membrane components affect membrane stability? So, we said that before, steroids such as cholesterol gives membranes more stability, meaning it doesn't let the membrane become too fluid or too static, too rigid. It keeps it in between. It gives it stability. That is the function of cholesterol in our plasma membrane, or in just a plasma membrane in general. Which of the following statements best describes a feature shared by mitochondria and the endoplasmic reticulum that increases the efficiency of their basic functions? No need even to read the upper part of the question here. You just need to read that. We both know that the mitochondria and the endoplasmic reticulum are highly folded. What does highly folded mean? It means that their surface area increases, and when surface area increases, efficiency of exchange increases. That means the thing that increases efficiency is surface area. And what increases surface area? The highly folded membranes. So what is the thing that increases the efficiency? It is the highly folded membranes that actually increase the surface area. So highly folded membranes. Based on the information provided, Gaucher disease results most directly from a defect in the function of which of the following organelles. So we can't conclude the answer from just that. We're gonna have to go up and read. So what is Gaucher disease? Gaucher disease is an inherited disorder in which cells of the body are unable to break down a particular type of lipid. Try and remember what's breaking down while I continue reading the question. Resulting in the buildup of the lipid in some tissues of organs. Now, what is the organelle that breaks up things? What is the organelle that digests things? It's the lysosome. So, if we have a trouble with digestion and breaking down particular types of anything, that means we have a defect in the lysosome. Number 27, which of the following best describes the process that occurs within the Golgi complex? Okay, so all eukaryotic cells contain at least one Golgi complex, typically located in the cytoplasm near the endoplasmic reticulum. Okay. What happens in the Golgi complex? Just like I said before, the enzymatic modification of newly synthesized integral membrane proteins. To put it into words, ER makes a protein. The protein is transported by the ER to the Golgi complex. The Golgi complex folds it, modifies it, adds maybe a carbohydrate chain, I don't know, it does something to it, packages it, makes it ready, and then off it goes into the world. Okay? That's what happens. So, what happens in the Golgi complex, specifically in the Golgi complex, enzymatic modification of newly synthesized integral membrane proteins. Number 28. Oh, for the love of God. Okay. Oh, thank God. So based on the data, which cell is would likely be most effective in the exchange of membrane materials? Here we don't have to read up. What is the thing that results in efficiency? Effective exchange. Effective exchange is the same as efficiency of exchange, same thing. So effective exchange means efficiency in exchange. What is the thing that is responsible to that? The surface area to volume ratio. The surface area to volume ratio has to be very big. So the bigger the surface area to volume ratio, the better it is at exchanging materials. Here we divide surface area by volume to get the ratio. The one with the biggest answer, which is 60 divided by 10, which is 6, is cell 2, and that is the most efficient. Which of the following provides an accurate calculation of the surface area to volume ratio of an HS red blood cell, as well as a prediction of its effect on the efficient transferring of oxygen compared to a normal red blood cell? So here, you guys should remember this from before because we had it in the last quiz. By the way, I didn't get all of these questions wrong. Before you say that the guy that's teaching me doesn't know it himself, I put any answer for all of this quiz. I was just selecting random answers so I can actually get the marking scheme and the correct answers. Okay, so don't worry. Anyways, continuing. Here we have the hereditary HS is a disorder of red blood cells that causes the cells to be smaller and spherical instead of having the usual flattened biconcave shape. The average diameter of the normal red blood cell is 7.2 micrometers, and the average diameter of red blood cells is with HS was found to be 6.7 micrometers. So the normal blood cells are basically bigger than the smaller blood cells, okay? Now the normal red blood cell has an average surface area of 136 and an average volume of 91. So they gave you the normal volume and the normal surface area of a normal red blood cell, okay? What you have to do is find the volume and the surface area of the abnormal blood cell, which is the HS blood cell. You already have the diameter. You divide it by 2 to get the you divide it by 2 to get the radius. Why do you need the radius? Because they told you here that the HS blood cell is spherical. How do you find the surface area and the volume of a sphere? Surface area of a sphere is 4 pi r squared. And the volume of a sphere is 4 over 3 pi r cubed. Surface area, volume. You find these two answers, you divide them by each other, you do surface area over volume, you get the ratio. And obviously your ratio will be smaller, so the ratio is 0.89. So the answer you get from dividing the surface area by the volume is 0.89. That means the cells are less efficient at transferring oxygen because the ratio is smaller. Now, which of the following best predicts what will happen to lysosomal enzymes if the proteins that transport H+ ions from the cytosol into the lysosome are damaged? Here, no need to read. H+ ions. What do H+ ions do? They make things more acidic. And what is the lysosome's main thing? It's digestion. How does it digest? By being acidic. It needs an acidic environment to digest things. If it doesn't have that acidic environment, it can't digest things. So without the H+ ions that give it its acidity, its pH will increase, making it less acidic, because you know how the pH scale works. The pH scale, if your pH increases, that's more basic. If the pH decreases, that's more acidic. So our pH will increase, and the lysosome won't be able to digest anymore. So it will become inactive. It will not become active. The lysosomal enzymes will not become active since there will be no active transport of H+ ions. Okay, which of the following best explains why hydrolytic enzymes are typically transported to the lysosome after they are synthesized by ribosomes? Why do we take enzymes to the lysosome? Why do we need that? Because these enzymes are essential for breaking down the organic molecules. The enzymes are the things that break down the organic molecules that give the lysosome the pH it requires, the acidic pH. That is why we transport it to the lysosome. We transport these hydrolytic enzymes that are essential for breaking down the organic molecules, which is the function of the lysosome itself. Now, which of the following outcomes will most likely result from a change in shape of the mitochondrial inner membrane from a highly folded surface to a smooth, flat surface? What is the mitochondria's job? The mitochondria's job is to create ATP, it's to make energy, cellular respiration. If it has a lower surface area, because we know that if it goes from highly folded like this to flat, the surface area decreases. And when surface area decreases, the surface area to volume ratio will also decrease, which is bad. That means it will become less efficient. Whatever, spelling doesn't matter. It will become less efficient. So, mitochondria will become less efficient because the surface area of the inner mitochondrial membranes will decrease. Which of the following statements accurately describes the facilitated diffusion of materials across a cell membrane? How does facilitated diffusion work? It's from higher concentration to lower concentration across a channel or carrier protein, a transport protein. T protein. Transport protein. Okay? Ions move down the concentration ions move down their concentration gradient across the membrane. Okay. Number 34. Which of the following outcomes will most likely result from a loss of ion pump function in the cell's lysosomes? Okay, we can't make out what's happening, so we're just gonna have to read it. The figure shows a process by which a cell might absorb food from its surrounding environment and break it down for use as a source of energy and matter. The process involves lysosomes, which are membrane-bound organelles that contain hydrolytic enzymes. Activation of the hydrolytic enzymes requires an acidic pH, and lysosomes maintain an internal acidic pH by using ion pumps. Okay, now we know what the ion pump does. The ion pump is the thing that maintains the acidic pH of the lysosome. Basically, it's the thing that is pumping out H+ ions to keep the acidity of the lysosome down as low as possible. We need it as acidic as possible, okay? That means the internal pH of the lysosome will then increase, which will prevent the activation of hydrolytic enzymes and interfere with the intracellular digestion of food. Number 35. Some viral infections can lead to the rupture of the lysosomal membrane. Which prediction of the effect of this disruption of cellular compartmentalization is most likely correct? So, what happens if the lysosome membrane breaks? All of those hydrolytic enzymes will be released, the acidity will go crazy, and the digestive enzymes will go out. That means the cell itself will then become digested. So the lysosome, the enzymes that were inside the lysosome, when they get released, they will digest the cell and kill it. It will cause cell death. So hydrolytic enzymes will be released, which will cause cell death. Sorry guys for my voice, by the way. I don't know what's going on with my throat, so my bad. Okay, so which of the following claims is scientifically accurate and consistent with an observation that a decrease in lysosome production within a cell leads to a decline in mitochondrial activity? Okay, so we know that the lysosome breaks down macromolecules, and for cellular respiration to occur, it needs those macromolecules. It needs the nutrients from the macromolecules. So if the lysosomes aren't working, that means, if there are fewer lysosomes, that means, Bismillah ar-Rahman ar-Rahim. Yeah, exactly. Sorry. Yeah, so if there are fewer lysosomes, that means that the macromolecules the availability of nutrients from the macromolecules that are being broken down will decrease. That means that the cellular respiration rate will also decrease. So fewer lysosomes will be available to break down macromolecules to provide the necessary nutrients for cellular respiration. So here we go, number 37. What is wrong with me? A cell's membrane potential is maintained by the movement of ions into and out of the cell. A model showing the influence of membrane proteins on the movement of sodium Na+ and potassium K+ ions across the barrier membrane is presented in figure 1. Okay, so here we have it again. So we have the high Na+ and the low K+ concentrations in the outside of the cell. So this is outside, this is inside. Okay, here we have the low Na+ and the high K+ concentrations inside the cell. Okay, now let's see what the question is asking us. Based on the model represented in figure 1, which of the following outcomes will most likely result from a loss of protein X? Obviously here, look here, what will happen. Protein X is the thing that is transporting K+ outside of the cell, meaning that it is increasing the K+ concentration outside of the cell, so it's making it higher and higher. That means that the concentration of K+ outside of the cell will go down if this protein isn't there anymore. So if this protein isn't there, K+ can't leak out, K+ can't go outside of the cell, meaning that the concentration of the K+ ions outside of the cell will go down, it decreases. And the membrane potential will be disrupted by an increase in K+ concentration inside the cell or a decrease of K+ concentration outside the cell. It's the same thing, just like what I said, okay? Increase of K+ inside the cell or a decrease outside the cell, it's the same thing, okay? If it were written, both would be accepted. But that was multiple choice, and I didn't get a good look at the option. So Based on their observations, what should the researchers do to further clarify how the availability of the protein outside of the cells affects the rate of endocytosis of the protein? Okay, we couldn't make out what's going on, so let's read. Researchers investigate the transport of a certain protein into cells by endocytosis. In an experiment, the researchers incubate the cells in the presence of the protein and measure the amount of the protein that is absorbed into the cells over a 5-minute period. Now, based on their observations, what should the researchers do to further clarify how the availability of the protein outside the cell affects the rate of endocytosis of the protein? Obviously, they should try incubating the cells with different concentrations of the protein. So you should do one with a little bit of a lower concentration and a little bit of a higher concentration and measure and see what goes on. Okay. Number 39, researchers exposed identical cells to solutions containing different solutes. Each solution had a higher concentration of the solute outside the cells than inside the cells. So all of them had a higher concentration of solute. Solute concentration was higher outside of the cells than it was inside of the cells. Now, the solutes varied in size, charge, and polarity. After a set time, the researchers measured the concentration of each solute inside the cells to determine the permeability of the cell membranes to each solute. Here are the results. Based on the data in Table 1, which of the following statements best supports the hypothesis that only certain types of molecules can move freely across the cell membranes? Look there. Small nonpolar solutes showed the greatest increase in internal concentration, while large nonpolar solutes showed little to no increase. So, what they're asking us is that only certain types of molecules can move freely across the cell membranes. And what are those certain types of molecules? The small nonpolar molecules. And as you can see here, they told us that all of the initial concentration outside of the cell was high and all of the concentration inside of the cell was low. So at the beginning, outside was high, inside was low. We want to see at the end which one inside was high now. And what's the only one with the high one? The small non-polar solutes, so solute A. So the small non-polar solute was the only one that had a high concentration inside of the cell. Excuse me. Meaning that it is the only one that can move freely across the cell. It is very easy. Coming in, going out, it's very easy for it. Okay? And yeah, small nonpolar solutes showed the greatest increase in internal concentration, while large nonpolar solutes showed little to no increase. That showcases that some solutes, some things, can actually move across the cell membrane very easily in comparison to other things. Which of the following observations best supports the model? So, the Anderson-Bound theory proposes a model for the evolution of mitochondria. According to the model, an ancestral eukaryote engulfed a small free-living prokaryote organism. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host. And what best supports this model? Mitochondria and some prokaryotes share similar metabolic reactions that produce ATP. So that both the mitochondria and the prokaryote are sharing similar metabolic reactions. That means that the mitochondria most probably came from something else, meaning a prokaryote, which shows that we evolved from a common ancestor. And there you have it. Number 41. Which of the following observations best supports the claim that mitochondria evolved from a once free-living prokaryote cells by the process of endocytosis? Very similar to the last question, mitochondria are supported by a double membrane. That also showcases that it was actually engulfed by something else, because if you have two membranes, that means you have to have been eaten by something else to gain that second membrane. Now, muscle cells have high ATP demands. Which of the following is a scientific claim about the structure of mitochondria? And muscle cells should be different than it is in the other cells because of the high energy demand of mitochondria. So, if mitochondria has a very high energy demand, that means, if the muscle cells have a very high energy demand, that means the mitochondria will have to create a lot of ATP. And to create a lot of ATP, you need a lot of exchange. And to have a lot of exchange, you need a very high surface area. And if you have a high surface area, that means you have a high surface area to volume ratio. That means you're very efficient. So what causes the increase in that surface area in the first place is the folding. So the more folds, the higher the surface area to volume ratio, the more efficient. So what should the mitochondrial cell in the muscle cell look like? It should be highly folded. So the inner membrane of mitochondria and muscle cells should have more folds to increase the surface area, allowing more ATP to be synthesized, because the muscle cell needs a lot of ATP. Number 44. The Na plus K plus pump requires ATP to move the ions from one side of a membrane to the other. Which of the following explains the role of ATP plays in the pump's activity? ATP is the thing that provides the energy to move substances across the concentration gradient. We know that, against the concentration gradient, against the concentration. Now, which of the following, if true about the drug, best supports the alternate hypothesis that the new drug will exhibit simple diffusion across plasma membrane? Okay, so let's read. A team of biologists develop a new drug, and one team member hypothesizes that the drug is incapable of freely passing across the plasma membrane and requires the help of membrane proteins to enter cells. Alternatively, another biologist on the team hypothesizes that the drug can diffuse passively across the plasma membrane, like O2 and CO2 can. So that's the alternative hypothesis. And what are O2 and CO2? They are small non-polar molecules. That means to support the alternative hypothesis, the drug has to be a small non-polar molecule as well. Now, the cell membrane is selectively permeable due to its structure. Thus, the internal environment of the cell is distinct from the external environment of the cell. One biologist hypothesized that small non-polar molecules readily pass through the membrane. Another biologist alternatively hypothesizes that the types of molecules require channel and transport proteins that are embedded in the membrane in order to move across the membrane. Which of the following data would refute this alternate hypothesis? So we want to prove the alternate hypothesis wrong. CO2 and N2 movement in and out of the cell is unaffected when membrane protein activity is blocked. So the alternative hypothesis is that everything needs a transport protein to enter the cell membrane. But to prove that wrong, they blocked the transport protein. So basically they blocked it, they made it ineffective. And then they saw that the CO2 and N2 movement in and out of the cells was unaffected. So if the CO2 and N2 movement inside and out of the cells was unaffected, that means that it was never relying on the transport proteins. That means that the alternative hypothesis is completely wrong, which is why we choose this option. And number 47, oh my god. Researchers studying osmoregulation in a specific crab species measured the change in water content within the crab's muscles when the crabs were exposed to water with different salt concentrations. Okay, whatever. Which of the following claims about the crab's muscle water content is most likely supported by the data shown in figure 1? So we're going to have to look at the data shown in figure 1, but we can also use our own understanding of this topic. So the water content decreases as water osmolarity increases. Okay. So if you have cells, okay, if you have cells with high water content, with water content, okay, and then you put it in a solution with high salt content, with a high solute content, meaning a high osmolarity. Osmolarity means solute concentration. A high osmolarity means a high solute concentration. So it has a high solute concentration, high osmolarity. Okay, what do you think is going to happen? So we can balance the external and internal environments. We need to remove some of the water that's inside the cell and put it and add it to the solution that is outside of the cell to make both water potentials equal. Okay, we want the water potentials of both the inside and outside of the cell to always be equal, meaning that the water content inside the muscle cell will decrease as the osmolarity out of the solution outside the cell increases. Exactly what they said here, okay? And you can just take it from the graph as you see here. The water osmolarity increases, the muscle water content decreases. The thing, that's it. What is this? How am I here? What? Number 47. So I have to go number 48. Okay. Testosterone movement across the cell membrane. Okay, so testosterone is a small steroid hormone that is important in cell signaling. So they told us that it is a small steroid hormone. Okay. Which of the following describes where testosterone enters a cell and why it is able to cross at that point? Okay. So they told us that testosterone, it is small, and it is a steroid hormone. Steroids are a type of lipid, and what are lipids? They are insoluble in water, meaning they are non-polar. And what do we know about small non-polar molecules? They pass through the cell membrane very easily by passive transport, meaning that they don't need any of this or this or this or anything. It just goes straight through. So number 1 is the correct option, because testosterone is non-polar and can diffuse through the membrane. It is small and it is non-polar. Why? Because it is a steroid, and all steroids are non-polar. Now, which of the following best describes the number areas? So they're saying that number 1 and 3, area number 1 and area number 3 are polar, and that's right because it is in line with the polar heads. It is in line with the polar heads. And area number 2 is non-polar because it is in line with the non-polar tails. In line with the non-polar tails. And that's it. But they didn't mention area number 2 in the answer. They just mentioned the areas number 1 and 3 are polar. That's it. Okay? There we go. Now, based on figure 1, which of the following membrane components are most likely non-polar? Obviously the phospholipid tails and the cholesterol. You guys don't even have to look at the diagram for it. You just go down here. Phospholipid heads are polar. The phospholipid heads are polar. The phospholipid tails are non-polar, but the embedded protein could be polar and non-polar at the same time, or just polar, or just non-polar, whatever. It can be both. Okay. The only thing that is always non-polar, the only things that are always non-polar, phospholipid tails and cholesterol, out of these options that we have here. Number 51, based on the model, what will be the mean diameter of the phytoplankton cells that are found 25 kilometers from the shore? Okay, basically they're saying here that you have a shore and then you have phytoplankton cells. Here the phytoplankton cell has a diameter of 900. Okay, and as you move away from the shore every 5 kilometers means that the phytoplankton cell's diameter is 50 less. Okay, so every 5 kilometers you go away, the diameter decreases by 50. What if you move 25 kilometers away? If you move 25 kilometers away, what happens then? So for every 5 kilometers, 50 decreases. So how much will it decrease for 25? You just do 25 divided by 5, which is 5, and then 5 times 50, 5 times 50, which is 250. It will decrease by 250. Okay? If it decreases by 250 and the original was 900, what is the new? 900 minus the 250, and you will get 650, and that is your answer right there. Moving on. Oh my god. Number 52. In an experiment, cells were isolated from an aquatic plant and suspended in pond water, a sucrose, sugar solution, or distilled water. All of the cells, when viewed under the microscope compared with the cells in the pond water, the cell in the sugar solution appeared shriveled. So distilled water appeared inflated. The cells. Okay. So which of the following statements best explains the observation resulted in figure 1? So from what you can see here, the one in the sugar solution is shriveling up. The one in the distilled water is expanding, and the one in the pond water, it's neutral, like we're fine. Okay. As you can see here, there was a net movement of water out of the cell, suspended in the sugar solution, because the sugar solution has a very high solute concentration. That means that to balance it out, the water has to go out of the cell and balance everything out. So there was a net movement of water outside of the cell. And then there was a net movement of water into the cell by the one suspended in distilled water, because when we had the cell in distilled water, the distilled water has a higher water potential than the cell. So to balance it out, the water will go inside the cell to make everything balanced. Okay, 53. Okay. Okay, here basically they're telling you how are you going to describe region 1 and region 2. Obviously you're going to say that region 1 is hydrophilic because it's aligned with the hydrophilic heads, and region 2 is hydrophobic because it's aligned with the hydrophobic tails. That's it. Region 1 is hydrophilic because it interacts with an aqueous environment, the hydrophobic heads. Region 2 is hydrophobic because it interacts with the interior environment, hydrophobic tails. Hydrophilic heads, sorry. Okay, so this is just another experiment. Which procedure should be done next to gather data needed to meet the scientist's objective? So let's go up and see. The transport of a substance across a plasma membrane of a specific organelle requires energy. The rate at which the transport takes place also depends on temperature. Okay, so depending on temperature. A scientist isolated the specific organelle and then used the following treatments to determine the conditions that result in the maximum transport. All treatments contained the extracted organelle were maintained at 25 degrees Celsius. Okay? The data from this experiment indicates that the maximal rate of transport of protein X at 25 degrees Celsius occurs at an ATP concentration of 1 micrometer per milliliter. Okay, so here they're telling us what should he do next. He should incubate samples containing 1 micrometer per milliliter of ATP at 4 temperatures other than the 25 degrees Celsius. Why? Because they said here that it's dependent on temperature. So he should change up the temperature to get an accurate result. Okay? So he'd incubate the sample with the one that has with the one that's most efficient, the one with the maximal rate of transport, which is the 1 micrometer per milliliter, and then he changes up the temperature. Okay? And yeah, so correct, this procedure combines the optimal ATP concentration. So he took the optimal ATP concentration. You always want the optimal ATP concentration for transport with data for transport rates at 4 other temperatures. So he's seeing the effect of the temperature, which is always good. Okay, number 55. In an experiment, researchers provided a radio-labeled amino acid to living plant cells. After 1 hour, the researchers determined the amount of the radio-labeled amino acid that was in each of several subcellular compartments. The results of the experiment are represented in the table. Which of the following conclusions about the radio-labeled amino acid is best supported by the results of the experiment? So as you see here, the relative amounts of radio-labeled amino acid were mostly found in the mitochondria. That means that the cell is mostly incorporated into proteins that regulate and manage the metabolic reactions, because that's what the mitochondria does. It manages the metabolic reactions. So that's most probably what what the cell was doing. Number 56, please. Thank you. A scientist studying the various prokaryotic and eukaryotic species found floating in a sample of water taken from a marine ecosystem. Which cellular component will be found in the widest range of organisms in the sample? What is found in both prokaryotes and eukaryotes? Ribosomes. Okay. Number 57, please. Okay. A certain type of specialized cell contains an unusually large amount of rough endoplasmic reticulum. What does the rough endoplasmic reticulum do? What does the rough endoplasmic reticulum do? Think while I continue reading. Which of the following functions is the cell type most likely specialized to perform? Okay, so what does the ER do? It produces proteins, transports the proteins, secretes them, whatever. What is this cell most likely to do? It is most likely to produce and transport proteins, secrete proteins. Okay. Oh my god. Yeah. The team wants to extend the research project. What should the team of students do next to obtain data that are more conclusive? So let's see what the hell they're doing. Intravenous IV therapy is used for fluid replacement in instances of dehydration in humans and other animals. One type of IV fluid is essentially a saltwater solution. To determine the best concentration for therapy in people, a team of students is researching the effect of solutions of different salt concentrations on red blood cells. The following observations were made from 3 different red blood cell samples viewed under a microscope. So 0.3% saline, the cell swells. 0.9% saline, cells unchanged. 1.5% saline, cells shrink. Saline means like salt, like salty. So if it's 0.3% salty, the cell swells. Why? Because it doesn't have that much salt in it, which means that it probably has a higher water potential than the inside of the cell, meaning to balance it out, the water has to go from the outside to the inside. So it will swell, the cell will become bigger. In 0.9% saline, it's like in the middle, so the cell isn't changed, it's like an isotonic environment, so we're fine. Okay, nothing will come out, nothing will come in, it's already balanced, the water potentials are already balanced. The 1.5% saline is very salty, so it's 1.5% salty, it has like a lot of salt. Meaning that, meaning that the water potential of the external environment is probably less than the internal environment. So to balance it out, the water will leave the cell and go outside to balance the water potentials out. Okay, so the team wants to extend the research project. What should the team of students do next to obtain more conclusive data? So how do they make their cells more accurate? They repeat the process with other salt concentrations. So they see, okay, what if I increase the salt concentration a little bit more? What if I decrease it a little bit more? What happens? Does the cell eventually burst if I go 0% saline, like pure water? And does the cell completely die out? Does it shrivel if I go like 3% saline, very salty? You know, they see what happens. Okay, that's how they make themselves more accurate, more conclusive. Okay, question number 59 is this monstrosity. Guys, here basically it's like an SAT English question. Like they're telling you what is happening in the graph. What do you see here in the graph? What is the thing that you see على طول? You see this straight line? You see the straight solid line? Increase after 24 hours. It has what? It has a 5 gram salt per kilogram. It has 5 grams of salt per kilogram of salt water. Okay? So the Na plus and the K plus ATPase activity increases. The K plus ATPase activity increases only after 24 hours. After 24 hours, at the lowest saltwater concentration. Which one did it increase only? In the lowest saltwater concentration. It didn't increase for the 35, it didn't increase for the 20, but it did increase for the 5, which is the lowest salt concentration. So the activity increased only after 24 hours of the smallest salt concentration. Number 60, I feel like I'm yapping too much here. Okay, this will come back to it at the end. It's an FRQ. Stomata are pores on the surface of the leaves and stems of plants that regulate gas exchange between the plants and the atmosphere. Researchers found that the stomata density of the leaves of a species of the plant changes as the concentration of CO2 in the atmosphere changes. When grown at 350 ppm CO2, the plant has an average density of 300 stomata per millimeter squared. When grown at 400 ppm CO2, the plant has an average density of 250 stomata per millimeter squared. Which of the following best describes how the ratio of the density of stomata per millimeter squared, per CO2 concentration ppm CO2, changes as the CO2 concentration increases? Okay, so what happens when the CO2 concentration is increasing? The ratio is decreasing from 0.86 to 0.63 because fewer stomata are needed at higher CO2 concentrations. As you can see here, the ratio of 300 stomata per millimeter per second to 350 is 0.86. So you're just dividing the ratio of stomata per millimeter squared to the ratio of the CO2 concentration, which is 0.86 for the first one. And then the ratio of the 250 per millimeter squared to the 400 ppm CO2 is 0.63. So when we increase the CO2, it went from 350 to 400. We also decreased the stomata per millimeter squared decreased. So the ratio became smaller. This reflects that fewer stomata are needed as the concentration of CO2 increases. So if as we increase CO2, the concentration of the stomata is decreasing, that means as CO2 increases, we need less stomata. That's it, okay? You don't really need to calculate the ratio, but since they were like putting the numbers in and trying to trick us with the numbers, they were using different numbers in each option. It is very, very, very preferable that you should just calculate the ratios. Okay, you do the stomata over over the CO2 concentration. Okay, just they told you what to divide by. Let me go back. If you see here, they told you what to divide by. So they said how the ratio of density per CO2. So density over CO2. Stomata density over CO2 concentration. That's it. Okay. Number 62. Which statement correctly indicates the cell that is able to more efficiently exchange materials with the external environment and provide a correct explanation? So a spherical bacteria cell has a radius of 3 micrometers. The human egg cell has a radius of 100 micrometers. What do you do here? You're going to have to, You're going to have to calculate the surface area and the volume. Find the surface area. You find the volume, you divide it by each other. The one with the bigger ratio, the one with the bigger ratio is the winner. Okay, it is the one that is more efficient. So here the bacterial cell has the greater surface area to volume ratio, so it is the one that is more, efficient, okay? And if I hope you guys remember the equations, it's for a sphere, 4 pi r squared for surface area, 4 over 3 pi r cubed for volume. Okay, so don't forget that's for sphere. Okay, so now they changed the shape of our cells. Now we have a cylinder and a rectangular prism, for the love of God. Why? I don't know. Heck. Okay, so of the two cells represented in the figure, which would likely be more effective at exchanging substances with the surrounding environment? So it would be cell A because it has the largest surface area to volume ratio. As you've seen throughout this quiz, when they ask you for more efficient, they're either talking about surface area to volume ratio or just the surface area. Most of the time it's surface area to volume ratio, okay? So start correlating it with each other. Efficiently, surface area to volume ratio, surface area. Start thinking about that, okay? You should have already been thinking like that. So if you haven't, you should start now. Okay, so we're gonna have to calculate the surface area and the volume. To calculate the surface area of this beautiful thing, you're gonna have to find the area of each of its faces. So you're gonna have to find the area of each of its faces and multiply it by each other. So it has two of these. So if this is 6, then this is also 6. So you're gonna have to do 2 into 4 times 6 because there are two faces. So 2 of these boxes means 2 faces. So you're gonna have to multiply it by 2. And then this face is 4 times 6. So 4 times 6. And then 2. The area of this is just 25 times 6. So 225 times 6 because it has 2 of these faces as well. And the area of this face is, and the area of all the faces are just length times width. So this is the length, this is the width. So 25 times 6. And for the two faces, you just multiply the answer by 2. And since we did this face and this face, now we just have this face left. This face is just 4 times 25. So we're going to do 225 times 4. We add these up all together and we have the surface area. Then to find the volume is just length times width times height for a rectangular prism. So you do this times that. So you do 4 times 25 times 6. So 4 times 25 times 6 and you get the volume. Okay, you add them, you get the surface area. And then you do surface area over volume, surface area over volume, and you get the ratio. And the ratio of cell A will be bigger than the ratio of cell B. Let me tell you how to find the ratio of cell B. The surface area of a what's it called? Ya Allah. The surface area of a cylinder is pi r squared h. Yeah, pi r squared h. Let me check. 1 second. So I was wrong. The volume of a cylinder is pi r squared h. Okay, the volume of a cylinder is pi r squared h. And then the surface area of a cylinder is 2 pi r squared plus 2 pi r h. Okay, please make sure you know this for Monday. Okay, then you're gonna just take the answer of the surface area divided by the answer of the volume and you will get the ratio. And then you're going to have to compare the ratio of this to this. The bigger ratio wins. Obviously, this will have the bigger ratio since we already saw that. And tada, that's the more efficient cell. Yay. Oh my God. Which of the following is a claim that could be made about the relationship between organelles 1, 2, and 3? Let's see. Organelles 1, 2, and 3. What do they all have in common? All 3 organelles play a direct role in production, modification, and transport of proteins that are released from the cell. Why, you're going to ask me? Yalla, I'll tell you why. Here we have the rough ER. Here we have the rough ER, okay? It will make the protein, transport the protein to the Golgi complex. Here we have the Golgi complex. Golgi complex. In the Golgi complex, it will modify and package the protein, and then it will send it to the plasma membrane. Send it to plasma membrane, where it will either go outside, or it won't even send it to the plasma membrane at all. It will go directly from here to like here, or from here to the lysosome, or from here to wherever, okay? It's either going to go inside the cell or outside the cell. If it goes outside the cell, it's going to have to go to the plasma membrane to be released. And there you have it. That is why they are all involved in the production, modification, and secretion of proteins. And transport. CD transport, not secretion, transport. Whatever. Based on figure 1, which of the following experiments would best confirm that protein A utilizes passive transport while protein B and C don't? What is the difference between passive transport and active transport? I'm not even going to read what's up. We compare the ATP consumption of the cells expressing each protein type during solute transport. Here, obviously, as you can see that there's solute transport going on here. Look what's going on here. Solute transport going on. Is there even anything to read? Yeah, there is. Okay. What's it called? Here, as you can see, there's solute transport going on. We want to see we want to see that if protein A utilizes passive transport and B and C don't. So what do? We see the ATP consumption of each. We just look at the ATP consumption of each. If A doesn't use any ATP, then we're good. That means it's correct. It uses passive transport. But if it does, that means no, it's active transport. But as you can clearly see here, for the protein A, high concentration to low concentration, that means we're good. It's passive. For this, well, it's not labeled, but I guess it's protein B because they look the same. Yeah, it's from low to high, active. So you need ATP, and then low to high, ATP. So just by comparing the ATP consumption, we can tell if protein A is using passive transport, if protein B is using passive or active, and if protein C is using passive or active. We can check if all of these are either using passive or active transport. Finally, last MCQ. Oh my god. Okay. Macrophages and B lymphocytes are cells that help fight infections. Macrophages engulf and destroy bacteria. B lymphocytes produce proteins that bind to bacteria in the blood. Based on the functions of macrophages and B lymphocytes, which of the following claims is most likely true of the organelles needed in both types of cells? What do we think? We need organelles that process and transport proteins because both cell types require an extensive endoplasmic reticulum and Golgi complex to process the proteins. The macrophages need these organelles for producing digestive enzymes, while the B lymphocytes need them for protein antibody production and secretion. Okay, that is why. What's it called? They need these organelles that transport and that process and transport proteins. Okay, because that's their job. They need the proteins to be either processed or transported to actually do their job. Because, Ya Allah. Okay, forget everything. Listen, macrophages and B lymphocytes are cells that help fight infections. Macrophages engulf and destroy bacteria. B lymphocytes, they produce proteins that bind to bacteria in the blood. Based on the functions of macrophages and B lymphocytes, which of the following claims is most likely true of the organelles needed in both types of the cells? So, let's go step by step. A, they need organelles that contain enzymes for digesting materials. Did they ever say, what's it called, that they're releasing enzymes or anything? No. So they release proteins, not enzymes. They need organelles that store macromolecules and water. Why would they need macromolecules and water? No. They need organelles that process and transport proteins. Yes, because the B lymphocytes are producing proteins that go and bind to the bacteria in the blood. And and they need organelles that produce lipids or for the plasma membrane. No, obviously not. What the hell do lipids have anything to do in this whole thing? Now, I know that some of you are going to come and tell me that, why don't you choose this one? Yes, I know that macrophages engulf and destroy bacteria. I get it. But do the B lymphocytes do that? No. They said they want something that is found in both. It is needed in both types of cells. And you need an organelle that can process and transport proteins in both of those cells for it to make it work. Okay? Okay, let's go to the FRQs. Oh my god, this video is so long. Okay. So, describe the importance of the inner membrane separating different regions of the mitochondria and the chloroplast. Okay, so part A. the response indicates that the inner membranes permit different reactions to occur in different regions of the organelles. So here they're just saying that what's the importance of compartmentalization basically in part A. The importance of compartmentalization is eliminating competing reactions. That what's it called? Basically, it eliminates like reactions happening at the same time that could affect the efficiency of the cell. So if I had like two reactions happening at the same time in the same place, they could mess each other up. That's why having membranes inside the cells, the inner membranes, is really good, okay? So we don't have things that compete with each other and mess each other up, okay? Part B, based on the diagram, describe how the structure of these organelles controls movement across the cell membrane. I personally went with surface area to volume ratio because we can say that, what's it called? I'd say that based on the diagram, it can be clearly seen that both cells have highly folded membranes, okay? And this therefore increases the surface area, which then therefore increases the surface area to volume ratio, which makes it more efficient at exchanging materials. And that gets you a mark. Go to part C. Complete the table to represent the relationship between volume and surface area of a spherical structure surrounded by a membrane. Here, basically what you're doing, you're just going to, You're just going to get the volume, 4 over 3 pi r cubed, surface area, 4 pi r squared. You just do the ratio, this divided by this, and we're good, we fill out the table. Okay. Part D, explain how the folding of the mitochondrial inner membrane and the many individual chloroplast thylakoids are beneficial to a cell that is carrying out chemical reactions. Part D, what have we been talking about the whole time? The response indicates that the folding increases the surface area. So the folding will increase the surface area. That means that the membrane will be able to hold more enzymes, do more reactions, and do more reactions that can take place than if less surface area was present. Okay, so what I tell you to say is I tell you guys to go with okay, I tell you guys to say the folding and the inner membranes, the folding of the inner membranes increases the cell surface area, which allows the cell to hold more enzymes, which speeds up chemical reactions. Rather than if we didn't have these foldings, the surface area would decrease, therefore decreasing the cell's ability to hold enzymes and decreasing the cell's ability to carry out chemical reactions. Okay, that's question number 1. Now for question number 2, let's go here. So describe how the phospholipids of a plasma membrane regulate the movement of large or polar molecules across the membrane. Explain how osmosis will affect animal cells when the cells are placed into an environment with a low water potential, high solute concentration, compared to the intracellular water potential. Okay. So here they're asking us 2 questions in one, basically. So let's read the given and see what's going on. So paramecia are single-celled aquatic eukaryotes that can survive in a wide range of salinities. Paramecia use contractile vacuoles to help maintain homeostasis under these varying salt conditions. The contractile vacuoles fill with fluid and then contract to remove the fluid from the paramecia. Okay? Scientists wish to determine the effect of salinity or contractile vacuole activity. They maintain different groups of paramecia in water of different salinities for 1 month. They then measured the average amount of fluid ejected from the contractile vacuoles in each of the paramecia each time the vacuoles contracted. So now they want us to describe how the phospholipids of a membrane regulate the movement of large or polar molecules across the membrane. What do we know about that? We know. So the response should indicate that the hydrophobic tails of the phospholipid fatty acids block the movement of large and polar molecules, which require protein channels to cross the membrane. Just like here, they're saying what is the effect of the phospholipids on the movement of larger polar molecules? We say that since the phospholipid center is composed of fatty acid tails that are hydrophobic, it will not allow of phospholipid. Sorry, guys. guys. Sorry, I know it's messy, just bear with me, okay? We say, for part A, for describing how the phospholipids of the plasma membrane regulate the movement of larger polar molecules, we say, since the phospholipid center is nonpolar, it will not allow the large or polar molecules to move across the membrane. Rather, it will force them to use channel proteins or carrier proteins, basically transport proteins, for them to actually be able to move across the membranes, okay, along those lines. Now we go to the second part of the question. They're asking us, explain how osmosis will affect animal cells when the cells are placed into an environment with a lower water potential. Okay, what will happen if we place an animal cell with no cell wall into an environment with a low water potential? It will shrink, it will shrivel, okay? What did they say here? The response indicates that osmosis will cause an overall movement out of the cell wall and the cells will shrink, okay? Just add the part where you say that due to the difference of water potential, the higher water potential inside the cell will then force the water inside the cell to go out, causing the cell to shrink, and that's it, okay? Here basically what you guys have to do to construct the table. So take this as a rule, okay? And by the way, since they're asking us to graph, you guys should get rulers, pencils, calculators, obviously everything. Get everything with you: pen, pencil, ruler, calculator on Monday, okay? Here you have the y-axis and here you're gonna have the x-axis. The x-axis is always independent, okay? Meaning it's the thing that we're changing. And the y-axis is always dependent, meaning it's the thing that we are measuring. What are we measuring here? Here we're measuring the average fluid output, and here we're changing the salinity of water. So this is the x-axis, this is the y-axis, okay? We plot these points. You guys choose, what's it called? You guys choose like a range, so choose each unit, like choose the unit differences between each block and block, okay? Y-axis put the average fluid output, and the x-axis put the salinity. Plot your points, draw a line, okay? And that's pretty much it. Just like connect the points with a line. Don't draw a line of best fit. Don't, like, if you had, like, for example, a point that looks like this, so a point that looks like this, so a point that looks like this, so a point that looks like, don't draw me a line that looks like that. No, draw a line that looks like this to this to this. That's what you draw, okay? Describe how increasing salinity affects the amount of fluid ejected each time a contractile vacuole contracts. Calculate the water potential of an animal cell without contractile vacuoles if water enters the cell and creates a solute potential of -2. Assume that the pressure potential is in the cell is 0. Okay. So we know that the perimetrial plants, okay, they take in the water and then they eject it. Okay. What causes them to take in the water? The thing that causes them to take in the water is the water potential, meaning that if the water potential is high here, higher than the inside, it's gonna come in, it's gonna go out, okay? So the higher the water potential, meaning the lower the salinity of the water, the more water it will eject, because the more water will come into the perimecia, that means the more water it will have to eject out of it. But if the water potential goes down, if the water potential goes down, meaning the salinity goes up, less water will go into the perimecia into the first place, meaning that less water will go out of the perimecia as well. So the more saline, the more salty, the more solute concentrated it is, the less it will absorb, the less it will release. Okay? That's how perimecia works. Now, to calculate the water potential, we say, what's it called? Water potential equals to solute potential plus the pressure potential. So it's just -2 plus 0 equals to -2. That's it. That's all you have to do. That's all you have to write. Okay. Let me just show you guys the marking scheme. So this is for part AI. This is for part AII. This is for part B. This is for part C. This is for part CII. Okay. And that's it. By the way, before you guys tell me it's 0 plus minus 2, for the record, minus 2 plus 0 and 0 plus minus 2 are the same, huh? So it doesn't matter like how you write the equation. As long as you write it, you're good. As long as you write pressure potential plus solute potential equals to water potential, you're fine. Okay, or water potential equals to pressure potential plus solute potential, and you can write anything. Like you can write this plus this equals to this, or this equals to this plus this, whatever. And as long as you write these three and the plus sign, you're fine, and an equal, obviously. Okay, let me show you guys the marking scheme for number 1 as well. So number 1A, number 1B, number 1C, and number 1D. Okay, then we go to number 3 now. Oh my god. Identify the most likely mode of transport across the membrane for substance L. Explain how information provided helps determine the most likely mode of transport. Okay, so let's go up and read. Scientists are testing substance L to determine how it enters mammalian cells in a culture. The cells maintain a 120 millimolar intracellular concentration. That means inside the cell, it's 120 millimolar concentration. Okay. The scientists determine the rate of entry of substance L into the cells at various external concentrations of substance L, 10 to 100. So this is the extracellular, this is the intracellular. That means the intracellular is more than the extracellular. That means what? For water to enter, because here, look, it's rate of entry of substance. For water to enter from outside to inside, it has to go from low to high. And from low to high, it needs ATP. And ATP means active transport. So when they asked us in part A to identify the most likely mode of transport, we say what? Active transport. Now, when they tell us to explain how the information provided helps us, we say that they told us that the intracellular is 120 and the values tested are between 10 and 100, meaning that the intracellular concentration is greater than the extracellular concentration, meaning that the substance L will have to move from an area of low concentration to an area of high concentration against its concentration gradient, meaning it will have meaning that it will need ATP, which means that it will have to undergo active transport. Okay, this is the marking scheme for part A and part AII. Guys, remember that for AP style questions you need like a proper structured paragraph, so make sure you have that, a proper structured paragraph. Okay, and yeah, just proper structured like answers. Does not a paragraph, but proper English. Yeah. For part B, here we're just graphing. Remember, the y-axis is dependent, the x-axis is the independent, the y-axis is the thing that we're measuring, the x-axis is the thing that we're changing. We go up here. This is what we're measuring, this is what we're changing. So this is the x-axis, this is the y-axis. Okay. And you just plot your points. You plot your points, whatever, point here, 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 whatever, okay? And then you just draw your line, okay? Remember, don't draw this for them. Connect your points. Please connect them like this, okay? And determine the external concentration of substance L that will result in 1/2 of the maximal entry rate. The maximal entry rate is what? 100%. As you can see here, this is the maximal entry rate. What's half of it is 50%, meaning that it's somewhere in between here. Okay? So any value of 30 and 40 should be accepted tomorrow. I don't know why they told us between 31 and 34. I'm not really sure, because the most accurate answer is supposed to be 33. I did it like this because if 30 gives you 45%, 50% will give you how much? Okay, and then I just cross multiplied, and then I found the question mark, which turned out to be 33 point something, whatever. Like you just rounds to 33. So I do it the way I did it, but I think they're going to be lenient with us and give us between 30 and 40, as long as like we know what's going on. Okay. Part C, determine the external concentration of substance L that will result in 1/2 of the maximum entry rate. Oh my god. How sleepy am I? Predict the likely effect on the ability of substance L to enter the cells if substance L is attached to a large protein instead of free in the culture. If substance L is now attached to a protein, do you think that it's going to be able to pass through the plasma membrane as easily as before? Definitely not. Meaning that your response should indicate that substance L will be unable to enter the cell, just like I said, if it is attached to a large protein, or it can enter the cell as long, substance L won't be able to enter the cell unless there is a specific cell membrane receptor for the protein that it is attached to. So I attach substance L to a protein. Either you say that the substance L attached to this protein can't enter the cell membrane, which is way simpler, okay? Or you can say that the substance L attached to this protein can't cross the cell membrane unless there is a specific receptor for this protein in the plasma membrane. That is the only way it can then pass through, okay? Question number something, because I don't know where that last effective number 60. Okay, so let's see. Part A. Describe the biological need for cells to be surrounded by a membrane that is selectively permeable for different materials. Guys, without reading, what do we think this is? The response indicates that a cell membrane separates the internal environment from the external environment, or that a cell that is selectively permeable can help to regulate what molecules move between the different environments inside and outside of the cell. Guys, what is the function of the plasma membrane? It is separating the internal from the external environment. That's all you have to write. That is all you have to write. That's all you have to indicate. As long as you have that in your response, you're fine. Okay? Now, part B. Explain how the model shows selective permeability of the membrane to specific ions. We go up here. We see. Look here. The small lipid molecules are going through. We aren't talking about that. We want the ions, specific water-soluble ions. Look here. These are the only ones that are passing through. Look, they're the gray ones, the colored in ones. These are the only ones that they're letting through. They aren't letting these ones through. Look, they aren't letting these ones through. That's how it shows the selective permeability. Okay, so part B. The response does not indicate that the response indicates that a membrane channel protein is selective for a specific kind of ion because only one of the two kinds of ions represented can pass through the channel. Maher, why is it a channel protein? Ya Jama'a, I drew it 20 times. It looks like this. It looks like that. It's a channel protein. Channel proteins are specific for one type of protein. One type of ion. That is the selective permeability. Okay. Part C. Describe the characteristics of the phospholipid bilayer that permit small hydrophobic lipid molecules to pass directly across the membrane. Oh, look there. Small lipid-soluble molecule directly across the membrane. It is the hydrophobic tails because they are similar. The response indicates that the interior of a phospholipid bilayer is hydrophobic, which allows a hydrophobic molecule to diffuse across the membrane. Small lipid-soluble molecule is hydrophobic. The interior of the phospholipid bilayer is also hydrophobic. They are similar. If they are similar, it will pass through easily. No problem, okay? So as long as your response indicates that the interior of a phospholipid bilayer is hydrophobic, meaning that it will allow the small hydrophobic molecule to diffuse across the membrane, we're good, okay? Part D. Based on the model, explain whether the molecules shown crossing the membrane require energy to do so. Look here. High concentration to low concentration, high concentration to low concentration. Obviously it will be passive transport, meaning that the response should indicate that energy is not required, because I said passive transport, for the substances to cross the membrane because the substances are being passively transported from a region of higher concentration to a region of lower concentration. Exactly what I said. Okay. Finally, that is all. This is the longest video I've made. Okay. I hope this helps. I know that I was a little bit messy صراحة, بس this quiz isn't as easy to explain as the others. It's a bit more complicated. So yeah, number 14, I genuinely don't understand it yet. I will try figuring it out and posting it as soon as possible. Okay. Guys, remember, study course revision, study the course practices. Even if you want me to do videos on the course practices, let me know. Do everything you can do for this periodic. Make sure you're ready for it, because like, it's important. You guys, especially like bio has a big toll on your averages, so be careful, be ready, make sure that like you're all ready. Okay. Thank you everyone, have a nice day, bye bye.